Next up, George Hoeferlin, improving brain implants by changing the gut microbiome.
Getting up out of bed, holding a loved one's hand, brushing your teeth. All actions that we take for granted as able-bodied individuals. But for the hundreds of thousands inflicted with spinal cord injury, this can be but a dream from the past. Thankfully, scientists, clinicians, and engineers have developed technologies capable of restoring function to these individuals and improving their lives, giving people like this man the ability to move and do tasks on his own for the first time in years. And beyond that, helping us better understand the brain to tackle other diseases. All these are possible due to the same technology, brain implants that interface with neurons. However, the problem in this field has remained the same. Constant inflammation due to our implant causes damage to both the brain and the device. And due to this chronic inflammation, brain implants usually only last for a few months to a few years, leading to a loss in rehabilitation for the patient. That's where my research comes into play. In my thesis, I explore different treatments to help reduce damage and inflammation in the brain with the goals of prolonging implant function in the lives of those who use them. And I believe the solutions to our problem may lie not in the brain, but in the gut. The gut microbiome is an environment consisting of trillions of bacteria all performing various functions. So you might be asking yourself, so what? Why would I care about bacteria in the gut when I'm looking at inflammation and implants all the way in the brain? Neural disorders like anxiety, depression, and even Alzheimer's disease have all been linked to changes in the gut microbiome, and my work has shown that brain implants are no exception. Even within a week of implantation, the gut microbiome of mice is significantly changed compared to a healthy non-implanted mouse. Somehow, an implant in the brain is causing changes to bacteria all the way in the gut. But is the opposite also true? Can changing the gut impact the brain? To explore this, I treated implanted mice with either antibiotics or probiotics to alter their gut microbiome. Doing so caused significant changes to the types of bacteria present in the brain, corresponding to our treatments. Additionally, antibiotic-treated animals resulted in significantly improved short-term function of our brain implants, while probiotics offered a more stable long-term improvement. These experiments suggest that changing the gut microbiome does in fact change the brain, and that doing so has huge impacts on how brain implants function. So my thesis so far has confirmed this connection. Now the questions are how and why. Are bacteria from the gut directly migrating to the brain that cause these changes, or are they somehow communicating via the nervous system? My next set of experiments will look at hundreds of proteins and thousands of genes to track down this specific mechanism. And if I can answer this question, an entire new approach for therapeutics becomes available for reducing inflammation in the brain and prolonging brain implant function. And with that, extending the improvements of life to those who need them. Thank you.